Good morning, everyone. Today we are going to discuss about body fluids and circulation, and the topics that we are going to cover are blood, the components of the blood, and the blood groups. Let us see what is blood. Blood is required for the transportation of all the materials by all the animals. Transportation of materials in animals are done by blood, lymph, and hemolymph in insects. The transported materials are nutrients, gases, metabolic waste, antibodies, and hormones. Blood, it is a special connective tissue consisting of fluid matrix, plasma, and foam elements. Now let us see what is plasma. Plasma, it is a straw-colored, viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water and 6 to 8% of it is protein. Proteins contained are fibrinogen, globulins, and albumins. Now let us see about the form elements. The formed elements are RBC, erythrocytes, WBC, leukocytes, and platelets, constituting nearly 45% of the blood. Now let us have a look at the comparative anatomy of the formed elements. RBC, they originated from red bone marrow, leukocytes from the lymph nodes and spleen, platelets that is from megakaryocytes. Lifespan of RBC is 120 days, leukocytes 13 to 20 days, and platelets 5 to 9 days. RBC non-nucleated, WBC nucleated, and platelets non-nucleated. The shape of RBC is biconcave, but WBC and platelets they are irregularly in shape. Count. RBC count is 5.5 billions per mm cube and WBC 6,000 to 8,000 per mm cube. Platelets count 1,50,000 to 3,50,000 per mm cube. Color of RBC is red in color due to presence of hemoglobin and leukocyte that is white in color. Platelets purplish in color. RBC and platelets they are one, but leukocytes can be again divided into granulocytes and agranulocytes. Granulocytes, white blood cells with secretory granules in its cytoplasm. Agranulocytes, white blood cells that have no distinct granules in their cytoplasm. Now let us see the division. WBC, granulocytes and agranulocytes. Granulocytes again can be divided as the neutrophils, eosinophils, and basophils. Agranulocytes as lymphocytes and monocytes. Now, let us discuss about the blood groups. The blood groups present in the human can be A, B, AB, and O blood groups. Now, let us discuss. ABO grouping is based on the presence or absence of two surface antigens, that is, a chemical that can induce immune response on the surface of RBCs, namely antigen A and B. Similarly, the plasma of different individual contains two natural antibodies, proteins produced in response to antigen. The distribution of antigens and antibodies in the four groups of blood A, B, AB, and O are as given below. Blood group A, antigen on RBC A, antibodies in plasma anti-B, donors group A and O. Blood group B, antigen on RBC B, antibody in plasma anti-A, donors group BO. Blood group AB, antigen on RBC A and B, antibodies in plasma that is nil, Donors group AB, A, B, and O. Blood group O, antigen on RBC nil, antibodies in plasma NTA and B. Donors group, that is O. Now, let us see the Rh blood grouping. The Rh antigen, similar to the one present in rhesus monkey, is also observed on the surface of RBC of majority, that is nearly 80% of humans. Such individual, they are called Rh positive person, and those in whom this antigen is absent are known as Rh negative. An Rh negative person, if exposed to Rh positive blood, will form a specific antibodies against the antigen, Rh antigen. Therefore, Rh group should be matched before transfusion of the blood. A special case of Rh incompatibility has been observed between the Rh negative blood of a pregnant mother with Rh positive of the fetus. The Rh antigen of the fetus do not get exposed to the Rh negative of the mother in the first pregnancy, as the two bodies were well separated by placenta. However, during the delivery of the first child, there is a possibility of exposure of maternal blood to the small amounts of the Rh positive blood from the fetus. In such cases, the mother starts preparing antibodies against Rh antigen in her blood. In case of her subsequent pregnancies, the Rh antibodies from the mother, that is Rh negative, can leak into the blood of the fetus, that is Rh positive, and destroy the fetal RBCs. These could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition, we call it erythroblastosis fetalis. These can be avoided by administering anti-Rh antibody to the mother immediately after the delivery of the first child. Hope you understand something from these classes. Thank you and have a nice day.